So the Western blot is a protein detection technique that separates proteins by gel electrophoresis, transfers them to a membrane, and uses antibody-based detection to identify specific proteins with high specificity. For the principle, so the proteins in a sample are first separated by size through the polyacrylamide gel electrophoresis. The proteins are then transferred onto a solid membrane, which is usually a nitrocellulose or a PVDF. The specific primary antibodies bind to the target protein on the membrane, and then a labeled secondary antibody allows for detection by chemiluminescence, colorimetric, or fluorescent methods. So how it works, so we have protein separation, so the sample proteins are separated based on molecular weight. For transfer, the proteins are transferred electrophoretically from the gel onto a membrane, and then blocking. Membrane is blocked to prevent nonspecific antibody binding, and then antibody incubation. So we have primary antibody will bind the target protein, and secondary antibody will bind the primary antibody. For detection, the signal from the labeled secondary antibody reveals the presence and quantity of the target protein. Applications of Western blot, it is used as a confirmatory diagnostic test, so it is actually the gold standard for confirming infections such as HIV and Lyme disease. It can be used for protein expression studies, so it can be used to measure the expression levels of proteins in research and clinical samples. It can also be used for post-translational modification analysis, so it can detect modifications like phosphorylation using specific antibodies. Can also be used for protein-protein interaction studies to be able to use to verify interactions in complex formation in molecular biology. Some advantages of the Western blot is it has a high specificity by detecting proteins based on size and antibody recognition. Quantitative and qualitative information about protein presence and abundance, and it is useful for detecting low abundance proteins in protein isoforms. Just some things to remember, Western blot combines gel electrophoresis and antibody detection to identify specific proteins. It is essential for confirming diagnosis and studying protein expression and modifications. Antibody specificity ensures accurate detection even in complex mixtures.